All right, so welcome. So we're going to have just like 8 days in person during this semester. Today we're going to actually do lecture for the first part of the morning. We'll continue talking about the chemistry, and then we'll take a break. And then we'll actually do that chemistry review lab. And I'll talk more about how the lab works once we get to the lab. I actually brought some examples of lab notebooks so you can get a better sense of how they tend to be laid out and kind of what the expectations are. And we'll actually get to do things at, adding carbohydrates versus electrolytes to water or oil. And again, we've talked about what happens. You all have expectations about what should happen, but I think it's useful to actually do it and just really get more of a hands-on experience that, what's going on with polar versus non-polar and all that, because it is going to be really important for the whole semester to have a good feel for that. Let me see if I can make this work. Whether or not we're doing this in person, but if we're online as well, we usually just start by going over that warm-up quiz. That warm-up quiz is kind of like a review of the main concepts. Hopefully this thing turns on. Hopefully you all appreciated the little cat eye on there. And the positive charge, the cutest eye over there. All right. And again, how do you get a cation? What results in a cation? Yeah, it'd be the loss of an electron. For instance, like in sodium or potassium or calcium or any of these things that are kind of on the left-hand side of the periodic table lose their valence electron or electrons, then you end up getting a net positive charge. Again, I think it is always interesting to look at the distribution. Like if you're up here, you're like, cool, I'm on it. If you do these little warm-up quizzes and you're finding yourself down there, it's not going to hurt you in the big picture because this is only worth like a fraction of a point, but it is worth paying attention to because it means that you're not getting the concepts at the deeper level that you're going to need that we continue building on. So again, if on these warm-up quizzes, you find you're not doing that well on one of them, that's just information that you got to go back and make sure that you review that material and really do get it. And we can just, this the feedback loop. This one is kind of going back actually to our very first day. Negative feedback loops, which are going to be happening constantly in this class in order to maintain homeostasis. We talked about a classic thing is just in temperature control. And your body temperature is falling, so your body responds by shivering to increase the temperature. Negative feedback loop. Some variable is changing, in this case, temperature is changing, and the response is going to work to oppose that change. So if your temperature is falling, the response is something that's going to try to bring the temperature back up. Right, so same thing, if the glucose level in your bloodstream is dropping, the response is going to be to release stored carbohydrate to bring the glucose back up. Anything like that, where you have some change and the response is to oppose the change. I saw somebody just walk in who just came in. What's your name again?
Natalia.
You're Natalia. All right, so the next one will be relevant for today. Non-polar molecules, how do they respond if you try to dissolve them in water? They don't dissolve. That's one of the take-home messages for our lab today as well. Non-polar, remember, like dissolves like. So if you take a non-polar molecule, for instance, like butter, or I should say, oils and fats and stuff like that, and you try to put them in water, you've got some basic intuition. They don't dissolve very well. And again, that's going to be really important because there's going to be important molecules in your body that are non-polar, all the lipids, the cholesterol, the fats, things like that, steroid hormones. They don't just go into solution when you put them like in the plasma of your blood. We're going to have special carriers and lipoproteins and things to carry non-polar molecules around in your body. They do need to go into the watery blood and move around, but they're not just going to dissolve like sugar or something. Separate individual water molecules are attracted to one another. The hydrogen bonds, those kind of electrostatic attractions between kind of more locally negative or more locally positive parts of individual molecules. If hydrogen bonds are kind of more weak, they tend to form, but then things move around, they break apart, they come back. These are going to be really important when we look at, just what does it mean when a neurotransmitter binds onto a receptor? You have serotonin comes into the neuron, binds to a serotonin receptor. It kind of, the lock and the key thing, but it's not like breaking off the key in the lock. It goes in there and then jiggles around and it breaks, it just separates and goes off again. So hydrogen bonds are going to be happening all the time, and they do kind of connect in, but then they can also unconnect. As opposed to covalent bonds. To break a covalent bond is a much more involved thing with chemical reactions and things that we'll be talking about today in lecture. It's also part of our lab today. What is going to conduct electricity in general? What kind of a solution? Yeah, basically anything that's an electrolyte, things that are ionically bonded. Ionically bonded would be table salt, NaCl. If you spent a little time getting ready for the lab today, you'll recognize calcium chloride as another typical ionically bonded compound.
Out of the two, which one is the correct answer?
So those are both correct.
Oh, because my marking wrong.
Mine too.
Because
Mine
it
too.
says choose all that are correct. That's what I got wrong too.
Yeah, it says choose all that
I didn't
choose
read
all
that.
that are. So you had to choose both of these for your 0.2 points. You only chose one, you got 0.1 points. So yeah, both of them are the correct answer. In these Canvas quizzes, there's a few basic forms of the questions we can use. Some of them are where you have one right answer, other ones are multiple right answers. So whenever there's more than one possible right answer, I'll make sure in the question I'll say, choose all that are correct versus choose one of them. Yeah, so calcium chloride, this is going to break apart into the calcium 2+ ions, cations, as well as the chloride ions. So this will conduct electricity. This also goes into ions, which will conduct electricity. Again, we're going to use both of these today in our lab, and we'll put them in water, and we're going to put the electrodes in and see whether or not it completes the circuit. So we'll see in a very kind of direct way whether or not this is true in the real world. Ionically bonded compounds such as sodium chloride, what happens to that molecule when it goes into water? It dissolves, but it also dissociates. It also, the two parts, the sodium and the chloride, separate into individual entities, which is going to be super important as we start talking about osmolarity. We're going to go into that much more today when we're getting into our continuation of chemistry review. So, let me try to get this out of the way so it's not in the way. All right, this is not good. Turn the computer off. It should disappear. All right, so we're going to continue with chemistry review. So far we've been talking about atoms and specifically about ions, which are the charged atoms, which are going to be super important. Then with the bonding, with ionic bonding, covalent bonding, create molecules. And what I'm going to talk about now are some of the basic kinds of reactions you should be aware of, where you can take either atoms and create molecules, or molecules and rearrange them into other kinds of molecules. Often releasing energy. Those are going to be really important chemical reactions we're going to be watching in this class. So let's think about chemical reactions. Where did all my pens up? Chemical reactions, and I'll often say like rxns, just whatever. When I was in school, that's how we kind of abbreviated it. So chemical reactions. Typically, there's some kind of reactants. Talk about going to some kind of products. One of the things to keep in mind is most of the reactions that we're going to be looking at are in some kind of chemical equilibrium. So it's important to understand this idea of the chemical equilibrium. You know, often we think of reactants going to products, but depending on the conditions, products can go back into the reactants and go back the other way. And as you know, if you increasing the concentration of the reactants, you'll push the reaction this way, and we'll have more of it, more products showing up. If we create more products and get rid of reactants, it'll actually move back this way. So conditions are going to affect where this thing ends up balancing. So this equilibrium is when things change, it's going to move and find some new equilibrium. So in fact, that's the way the buffers are going to work in our lab today. There's going to be either the dissociation or reassociation of the hydrogens and and the weak acids and bases and stuff like that. So this is important to remember, is we're usually not just doing a one-way reaction. Sometimes we are, but sometimes, a lot of times, we're looking at an equilibrium. What kind of things affect the rate of a reaction? Say it again? Temperature. Temperature makes a big difference. Temperature is just the energy that little atoms and molecules have. So if they're jiggling around more, they're more likely to ram into each other and either create something new or break apart. So the higher the temperature, the faster the reaction. You know, in industrial settings where you're trying to speed up reactions, you create these like reaction containers and keep things up really hot, and it really speeds things up. You get lots of stuff made. You know, in your body, that's not so practical. You know, in fact, you know, homeostasis for your body temperature is like 97 something degrees Fahrenheit. You know, it's not going to be that practical to heat everything up to make reactions go faster. What are other ways we have to speed up reactions? pH, not necessarily. We're going to see pH will affect the reaction level, the reaction rates indirectly through their effects on another major player in reaction rate. So pH is going to be really critical in this discussion, but not directly. But yeah.
Pressure?
What's that?
Pressure.
Pressure. So pressure would be like, and that's the kind of basic concentration. It would be like, you know, how much of a certain gas you have in a thing, but in general, the concentration. If you have more things here, it's going to tend to run this way. If you have more things here, it's going to tend to run this way, just because it's more likely that things are going to ram into each other and happen.
Presence of enzymes.
So that's a huge one, the idea of enzymes. This is where we're going to get to the idea of pH and all that. So let's talk about so enzymes. What is an enzyme? Yeah,
Catalyst, something that speeds up.
yeah, so it's an organic catalyst. Organic meaning just carbon containing. And catalyst has two properties. One is it speeds up the reaction. Or you can also say lowers the activation energy, makes it more likely that it's going to run in the current conditions. What is the other defining quality of a catalyst?
Not used up in the reaction.
Yeah, it's not used up. So it does its thing, speeds it up, but it's now waiting, ready to go again. So both of these are going to be important. Again, we're going to have a whole lab just looking at enzymes a little later and how enzymes function and their kind of dynamics. Let me talk a little bit more about enzymes. Let's say, let me get, what's your,
Sorry, Jeannie. I
It's
have my
Jeannie.
thing.
yes, it's okay. You can just have a seat. So, enzymes. Enzymes are typically a type of protein. You know, they've now found there are other kinds of molecules that can act as enzymes, as RNAs that can act as enzymes. But pretty much any enzyme we care about in this class is a protein. We'll talk more about proteins as the day goes on. Basically, we have some big molecule that is my enzyme. A big protein. We'll talk about again more about proteins in a little bit. There is some little area which we call like the active site, the part that's actually gonna be able to bind with whatever it actually helps facilitate. And let's say we have an enzyme whose job is to break apart table sugar into simpler sugars. Like, table sugar, sucrose is a disaccharide. Table sugar it's like a glucose and a fructose together. And we want to break it apart into two, but it won't break apart on its own easily. So there's an active site here on this enzyme that's job is to break apart my little sucrose, my little disaccharide. The thing that the enzyme binds to and works with is called the substrate of that enzyme. So let's say this is example. Here's some sucrose that my enzyme is going to break apart. The idea of binding, we'll talk more about, but binding is this kind of temporary bond, kind of like these hydrogen bonds. And you have this active site, which has just the right shape to fit our substrate. So an enzyme is very specific to a particular substrate. So my enzyme, here I have my substrate here. Here I have my enzyme. I'm going to end up getting what's called the enzyme substrate complex, where in this point, my substrate is making this binding in this active site of the enzyme. And there's something about settling into this active site that in this case is kind of tweaking the sucrose in such a way that it's going to break apart. Sometimes these are about assembling things. It might be taking two things and holding them just a way that they end up connecting together. But there is this is kind of where the magic is happening as the enzyme substrate complex forms. It helps facilitate the reaction on the substrate here. And then when we come out the other side, the enzyme is still just sitting there as it was. Enzyme with its little active site is still there waiting for the next go round. But now I kind of got my products. So I'm being a little messy here, but you can kind of get this idea. You have some substrate, something that is going to be acted upon. The enzyme, which is this big protein, this big molecule with a specifically shaped active site that just matches to bind the substrate. They join up. There is this enzyme substrate complex where we actually catalyze the reaction. And then after that, we have the products drifting off to go off to their destiny. And the enzyme is sitting here unadulterated, ready to go for the next round. So speeds up the reaction, not used up. This is where, remind me your name one more time? Ethan. This is where we get to Ethan's point about pH being critical for controlling reaction rate. Like the only reason this enzyme works is because the active site perfectly matches its particular substrate. So if there's anything that messes up the shape of that active site, it means you're not going to get this binding. You're not going to create this enzyme substrate complex. So if there is no binding, then there's no catalyzing the reaction. And pH is critical for enzymes maintaining their shape. In a little while later, we're going to talk about things that denature proteins, things that mess up the shape of proteins. And pH is one of the big things that can mess up the shape of a protein, nature of protein. So that's like one of the reasons why maintaining pH homeostasis is so critical in your body. Because if pH is off, the enzymes are now denatured, no longer have the right shape, and now all of these chemical reactions are no longer going to be functioning properly. So somebody just walked in who just came in. What's your name? Say it louder. Yeah. Okay. Okay. Yeah. So does that make sense? Uh-huh.
So if you're looking at that at a molecular level, you know exactly what's going on when an enzyme binds to the substrate. Like we can, in this instance, you would see how the glucose and the fructose are separating out.
Yeah. Yeah. There's like theories exactly how some of these work. But its basic idea is like in this case, as the substrate settles in and gets pulled into the active site, it tweaks the bonds within the substrate in such a way that it allows it to break apart. If your hand up, or you just kind of
Just resting.
okay, let's see. We will be spending lots of time with enzymes. In fact, I might as well just bring in the idea of most of the chemistry in the body does not happen in one step. Most of it is not like reactants going to product. Most of it takes multiple steps, and each step is going to be catalyzed by specific enzymes. So let me talk about that. Before this word binding, so let me just write that word specifically because it's a good word to remember. The substrate binds enzyme. That word binding is going to come up a lot. It's also going to come up when we look at, again, neurotransmitters binding receptors, or when we look at hormones binding receptors. Binding is this place where it's this kind of lock and key thing, where these two things have this kind of complementary shape as well as chemical properties so that they fit in, they hold together. But it's kind of a temporary thing where something happens and then it comes apart again. So binding will be a recurring theme in our class. Again, as we go on in the coming weeks, I'll give you some very explicit details of what binding might look like. Like, what does it mean when serotonin binds to a serotonin receptor on your neuron? Again, it's going to be both the physical shape that fits the molecule as well as the chemical properties, like making hydrogen bonds or little non-polar pockets and things like that. So there's a complementarity of both of the shape as well as the chemical properties that make the two halves kind of fit together. All right. All right, so let's talk a little bit more about kind of types of reactions and then this idea of metabolic pathways. What are some of the basic types? Decomposition, breaking things apart. So you have like some AB and it becomes an A plus a B. Decomposition. It's also called metabolism. You know, that's going to be breaking apart the sugar to make energy in your cells. So we see a lot of breaking things apart. What are other kinds of classes? Yeah, exactly. Take an A plus a B and turn it into an AP. This is also known as anabolism. I realized that it's been so long. We probably should do the hands thing because then I feel like people are trying to be polite and then they're getting like drowned out by the people who are excited to actually. So what are a couple of other ones? And I apologize if you've been raising your hand and I've been like ignoring you. I'm still getting my feedback here as well. What are other? There's a couple of other ones we should mention.
Dehydration.
Say it again.
Dehydration.
So that is going to be a specific version of something. We'll talk about that.
Combustion.
Combustion is also going to be like a specific thing about this decomposition.
Isn't there like a replacement?
Yeah, kind of that exchange or displacement. You have like an AB plus a C goes to A plus a BC. You know, we'll see this quite a bit. We'll often phosphorylate things. We want to add a phosphate group onto something. That's going to be a really common way to like turn an enzyme on or off. You know, so if you want to phosphorylate, you want to add a phosphate group to something, you can take an ATP. ATP is just adenosine with 3 phosphates on it. And this, along with some enzyme. And we can do this kind of displacement reaction. We'll talk about a specific kind of enzyme known as a kinase. That's job is to swap out phosphate groups. And we'll end up with ADP. Adenosine that only has 2 phosphates left. And we'll have the enzyme with a phosphate group hanging on it. Again, we're going to see this a lot. This is going to be a way to turn enzymes on or off, or open and close channels or things. Often phosphorylating things is going to be a common theme. But here we've done this kind of exchange. We took a phosphate that was on the ATP and just swapped it over to this other thing. In the case, almost all the reactions we're going to be seeing in our class, there'll be some enzyme whose job is to make that reaction go. In the case of this, we'll have some kind of kinase. You know, and this is how you usually do it. You usually have the enzyme kind of drawn in as coming in, but coming out. Because remember, it starts and ends, you know, unchanged. But in the process, it helped the reaction run. You know, we're going to see lots of different kinds of kinases. The first step in glycolysis is going to be a hexokinase that phosphorylates the glucose. We'll see kinases in using creatine phosphate to get lots of ATP fast and muscle contraction. We'll see kinases that are going to be turning on enzymes as part of signal transduction as we get into what's going on inside the cells in more detail. We'll see lots of different types of kinases, but this word kinase is kind of the generic term for an enzyme that is catalyzing a reaction that helps phosphorylate things. Somebody had their hand up back here?
Oh, I was just, you said kinase enzyme start and end unchanged, right?
So,
You said it starts and ends unchanged?
Yeah, any enzyme will always start and end as it was originally, and by definition of a catalyst. So exchange or displacement, these will be common as well. And the last kind are just the redox reactions. Oxidation reduction. And what's happening in redox reactions? Nikki?
There's a gain and a loss of an electron.
Yeah, there's gain and loss of electrons. Which one is losing? Which one is gaining?
Oxidation is a loss in electrons. And then
Yeah, so exactly. Oxidation is the loss, reduction is the gaining. Again, the kind of non-intuitive thing, it's you're reducing charge if you gain electrons. Most of the energy reactions that we're going to see, like in the mitochondria and stuff, are going to be versions of this. We're going to spend a lot of time. We'll spend quite a bit of time looking at electrons that are being moved from one molecule to another and releasing energy in the process. You know, you can use kind of the analogy of gravity, like potential energy. Like this box has potential energy with respect to gravity. If I hold it up really high, it's got a lot of potential energy with respect to the floor. And I can drop it and catch it here. Yeah, drop it and catch it here. You know, it gets to release some energy as it falls. And I catch it here. It's still got energy with respect to, I can drop it again and catch it here. And each time I hand it off from a higher to a lower place, some of that energy that was potential gets converted into like this kinetic energy. It's kind of the same thing in the molecules with how the electrons are configured with respect to the rest of the molecule. Again, an example that we'll get to see pretty soon, like NADH is a high energy electron carrier. So
It's a redox reaction.
yeah, this will be exactly. So we can take this NAD plus and we reduce it by adding 2 electrons to create these NADHs. This, you know, carries high energy electrons. Now, NADH, the electrons are like really up high. So when NADH then releases those electrons and gives them to something else, they usually end up at a lower energy state, and that release of energy as they fall from the high to the lower energy state can get captured to do stuff. You know, ultimately, it's going to be an important part of making ATP in the mitochondria. So NAD, this is just one of the right? Niacin. Niacin is a precursor to making NAD. So we're going to talk about it in more detail when we get into cellular respiration. But this idea that you can have high energy electrons, which you can then hand off to other molecules that hold them in a lower energy configuration. And in that handoff, energy is released and captured and used for practical purposes in the cell. So yeah, we'll meet NADH and FADH2 a little later in the mitochondria. So redox. Yeah, one of the weird things in these biological reactions is, you kind of look for the hydrogens to see where the electrons are, which is a little non-intuitive, right? The NADH is the actual, what actually matters here is not that there's an H, it's that it actually has high energy electrons that it can donate to something. And the NAD plus is the thing that is giving them up and is ready to get recharged if it's got an appropriate source. Does that make sense? And we'll come back and we'll meet NADH in much more detail when we get into the mitochondria. All right. So now what I want to do is talk about the idea of a metabolic pathway. So it's, speaking of cellular respiration, what is the basic chemical reaction that describes the breakdown of sugar to make ATP in your cells? Everybody read that? I know you've all seen that in Bio 110. Anybody remember that?
Glycolysis? Not glycolysis.
Glycolysis is the first part of it, for sure.
For the Krebs cycle?
That's part of it as well. But there's like kind of a overarching. What do we start with in cellular respiration?
Okay,
We
John.
start with glucose, C6H3, and C6, 12O6. That's glucose. What else do we need for this to run?
Energy?
Oxygen. Oxygen. Plus 6 oxygens. And what do we end up with? It's all over.
Oh, is that for the synthesis?
Photosynthesis is running this backwards, making the sugar.
Okay.
CO2. Yeah, we end up with carbon dioxide
And water.
and water. And the kind of goal, the thing that we got doing this whole thing for in the first place,
ATP.
lots of energy. If you're doing it in the mitochondria, you're capturing that energy as ATP. So this is the combustion of glucose. If you light sugar on fire, it's the same equation, except the energy is going to be light and heat as you warm yourself by the burning sugar. So this looks very simple. You know, taking sugar and oxygen, you end up with carbon dioxide, water, and energy. The reality is we're going to spend an entire lecture just looking at how this equation actually happens in the cell. It does not happen in a direct way like this at all. It happens in many, many kind of sub steps. So like all of these reactions, the sum total of all the reactions in your body we call metabolism. When we talk about, use that word metabolism, it's just referring to all of the reactions that are going on, all of these, catabolic, breaking things apart, anabolic, building things up, releasing energy, this and that. For this particular thing, it starts with glycolysis. It starts with the first steps. You start with sugar. The first step is not combining with oxygen, does not have anything to do with carbon dioxide or H2O. It's actually using a kinase to phosphorylate the glucose. So the first step is going to be making glucose with a little phosphate hanging off it. And we're going to use a it just means a kinase that phosphorylates a 6-carbon sugar. So the first step of this is going to be making phosphorylated glucose. And just to get through to the end of glycolysis, which is the first parts of this, we're going to go into step 3, into step 4, ... into step 10. This is going to be the end of glycolysis. And each step here, we're going to have some other enzyme that takes us from one step to the other. Here, this glucose is supposed to be at step 2. Each of these molecules that's getting made along the way that is not your final product, we call an intermediate product. So intermediates are these things that get made along the way, but aren't your final endpoint. And again, each of these steps is catalyzed by another enzyme. There's going to be another enzyme. Let me try to draw it better. Or enzyme, whatever. So each step is catalyzed by an enzyme. Each step makes some new product, but that's not your final product. That we call an intermediate. In the case of cellular respiration, this continues on into the Krebs cycle, and there's even more steps. And finally, at the very end, you get ATP. You know, let's say it's like, I don't know, step, who knows, whatever. We're going to see it takes many, many, many steps to get to the ATP by the end. But this is this idea of what we call a metabolic pathway. A metabolic pathway is these many steps that have intermediates along the way, have enzymes that take you from one intermediate to the next intermediate. And you start with your reactants, you end with your products, but it is not a, just a single hop. It's many little sub-steps. The nice thing about these is it gives you a lot of options to kind of control things or change things around. Like for instance, let's say you have enough ATP already. What happens if you keep eating Snickers bars and you already have more than enough energy for your body's needs? Say it again?
Yeah, you store it.
You store it. You store it as fat usually. So there's going to be off ramps on these metabolic pathways. With some other steps, instead of going to 11 or maybe go to 11 prime and 12 prime and and end up with fat. So you'll have making fat instead of, you starting with the sugar, but because you don't need ATP, you can divert this pathway and go off an off ramp and ultimately make fat. Or maybe make cholesterol. You need cholesterol actually for making your steroid hormones or making your cell membranes. So that's one of the things you can do with metabolic pathways. You can have, it can go this way, or depending on the body's needs and conditions, it might take an off ramp and go on to another metabolic pathway, although it had the same start. Other things, if you need ATP, it's not just sugar that can be broken down for ATP. What else can be broken down for ATP? Lipids, yeah, fats. You can break, you store fats to store energy. You can break down fats to release energy. So there might be other on-ramps It could be, fats, maybe proteins. You can also break down proteins. And the first steps are going to be unique. But eventually it's going to merge and the final steps will be the same. So you can have different starting places, but then merging to a common endpoint in a metabolic pathway, so different on-ramps onto this pathway. And if we're starting breaking down fats or proteins, the first steps are going to be unique to the fats or proteins, but the final steps are going to be the same no matter what we started with. And again, the same thing, these off-ramps. If I'm going to break down glucose, the first steps are always the same, but then if I'm going to end up making ATP versus fats or cholesterol, it's going to be different final steps. So I'll have some off-ramp where I go and change what the metabolic pathway looks like. What is going to determine, like, let's say I'm at this critical juncture here where I'm trying to decide, okay, the cell has to decide whether or not to continue going towards ATP or going to make fat. What is ultimately going to drive that branch point?
What enzymes are present?
Yeah, exactly. Which enzymes are present or are active? So enzymes are going to be turned on or off in your body depending on the body's needs. And if I'm at this particular intermediate, depending on what enzyme is active, it's either going to catalyze the next step over towards ATP or it's going to catalyze the next step down towards fat. So part of the control of cellular activities is going to be turning enzymes on or off in the cell. And that is going to influence which metabolic pathways are activated. Like that little thing I was talking about earlier about using phosphorylating things to turn enzymes on or off. That's one of the ways your cell has to turn enzymes on or off and therefore turn on or off specific metabolic pathways inside the cell. So, if we were in a biochemistry class, we would just spend 4 months looking at all the different metabolic pathways that are going on in your body. This is not that. So we're going to look in a relative amount of detail at cellular respiration, what's going on in the mitochondria, just to kind of give you a sense of this really critical pathway that is common to all your cells. And we will reference some of the other ones as appropriate as we're going through the semester. But you should be aware of this idea of metabolic pathways. And in fact, here, let me change this around. To okay. Right. Turn on when you turn off when you don't want it. While this is going, I'm going to find this thing for you. Okay. It's so good. All right. So basically what I kind of want to give you a sense of how just ridiculously complicated it all gets. Let me get a higher resolution one. Okay, I am. Let's let this do it. Okay, make this similar. Right, so in this kind of thing, it just kind of like what I've just been talking about with like glucose. Where's glucose in this thing? Hold on. Glucose is starting. There it is. It's kind of starting up here, going down the Krebs cycle. And you can see it is just in this thing, each little circle is an intermediate. Is some molecule that's made along the way in some metabolic pathway. These are all the molecules that are made in the breakdown of glucose and to make the ATP. Everything else here is yet another metabolic pathway with some intermediates. Every little line connecting one dot to another is part of the pathway that has its own unique enzyme to catalyze that step from one intermediate to the next intermediate. So just to give you a sense of how kind of complicated it is, how many on-ramps and off-ramps there are on all of these pathways. Again, in our class, we're not going to get into most of these, but I think it is important to understand the basic idea of what metabolism really is. It's all these chemical reactions. They happen in multiple steps. Each step is catalyzed by enzymes. Often there are options on how that pathway runs. It can have different outcomes depending on the body's needs, even if it has the same starting point. It can have different starting points, like start with fats or proteins or sugars, but all end up at the same place, making ATP. So just kind of putting that out there. The chemical reactions in the body are not just like the ones you saw in chemistry where you have reactants and products. There's multiple steps, and each step is catalyzed by specific enzymes that are unique to the particular substrates. Okay. Okay. That. A few more things. One good thing about using the little iPad to do the lectures is you just click on it. It's just blank. I forgot about what a pain in the butt it is to erase. You can shape it. What's that? Keeps you in shape. It keeps you in shape. It keeps, yeah, no. Actually, it's really nice to me. I guess you guys are sitting down like normal, but I'm not used to sitting down while I'm teaching. It's been horrible.
You like online learning? Do you like teaching online?
No. Yeah, no, there's some things it's been interesting having to integrate Canvas more into the class. And there is some stuff that is good about that. But in the big picture, no. So let's talk a bit about specifying concentrations and solutions and mixtures and all that, because that's going to be important. It's on a practical level as we go through the class. We talked about this a little bit in class last week. The idea of you have some kind of solvent, it's whatever is the majority. In general, the solvent for our class is almost always going to be water. Pretty much everything in this class is in water. The solute is the thing in less amount that's sitting in the solvent. And there's three main versions of these mixtures where you have a solute and a solvent. One is a true solution. This is going to be things like sugar water or salt water. This is where you have the solute totally goes in, and once it's in, it never falls out of the solution. So a true solution never settles. You can have a glass of sugar water on your nightstand, and a month later, it's still just a glass of sugar water on your nightstand. You don't have the sugar just end up at the bottom of the glass. Doesn't scatter light. That means that when you look through it, it looks kind of like glass. It might have a color even, but it doesn't look milky or anything. So true solutions. Most of the stuff we're going to see is going to be this. We also have what are called colloids. These are usually larger solute particles. So we still have the condition where the solute never settles. But you do have scattering of light. They tend to look milky. Colloids are actually going to be important to some degree as well. Like when you have emulsions of like kind of little, we'll talk about this more. Like actually the cytosol in a cell is a colloid. We're not going to spend a lot of time going into details of colloids, but you should know what they are. They have interesting properties too, where they can go back and forth between being more liquidy and being more gelatinous and stuff like that. In our lab today, just to kind of give you a sense of the difference on a physical level of what a colloid looks like versus a true solution, we have like a bottle of glucose. I can just. The starch, I don't think the starch, maybe I'll have, depending on how much time I have, I'll make some more up. But like if you have a sugar water, you all know from your basic, life experiences that sugar water is pretty clear looking. You don't really see much difference between looking at sugar water versus looking at regular water. It's not till you sip it that you know it's sugar water. Starch is a much larger molecule. Starch is a bunch of glucoses in big chains. It's a lot harder to get it into solution. You got to really spend a lot of time and warm it up, but eventually it will go into solution. The starch will not settle out. It'll stay this starch solution, but if you look at it, it looks kind of cloudy or milky. And that's how you know it's a colloid rather than a just true solution. Those bottles there do not illustrate it well because I think starch is trickier to get into solution. So I think probably when it was made, it wasn't adequately mixed up. All right. The last one, suspensions. Suspension is when you have much larger solutes that actually feel gravity enough to get pulled down to the bottom. So this would be like if you've ever taken like muddy water. You have muddy water and it looks all muddy, but then you put it in a jar and sitting on a desk, and the next day you have clear water with kind of silt sitting on the bottom of the jar. So suspensions, the solute does settle out. A classic example of a suspension in your body is your blood. The blood cells are big enough that if you just let them sit, they'll all go to the bottom and the plasma will be at the top. Your blood is usually just moving around, so everything stays mixed up. Later on, one of the labs that we will be doing in person is the blood lab, where we're going to look at hematocrit. We want to see the proportion of cells versus plasma in your blood. We'll take a sample of your blood. We can actually increase gravity by putting it in a centrifuge, basically just increasing the force of gravity. All the blood cells will get pulled to the bottom. The plasma will be on the top, and we can just see what is the ratio of plasma to cells. That only works because blood cells are big enough to get pulled to the bottom by gravity. So like blood is a suspension. So most of the things we're going to see are going to be in solution. Let's talk about specifying the concentration of a solution. All right. What what's you fall down in chemistry. What's like the main way you specify the concentration? Molarity. Molarity is going to be one of our main ones. Molarity. So molarity. This is tricky. I'm seeing myself. The molarity is moles of solute per liter of solution. So we have to make sure we remember what moles are. I've mentioned what does it mean to have a mole of something?
I have to do some liquid 6 times 10 to the 23rd.
Like 0.022 times 10 to the 23rd atoms or molecules of something. It's an amount of stuff. People use like the analogy of like if you're buying eggs at the store, a typical amount of eggs is a dozen eggs. So you get a dozen eggs. It's like a carton of eggs. It's a nice amount of eggs. Atoms and molecules are way, way dinkier. So if you want like a handful of glucose, you need way more than a dozen. You need like this many of the glucose molecules to have a handful of glucose. Yeah, this is 10 to the 23rd. This is is that many atoms or molecules or something. It's a whole lot, but they're all like submicroscopic. So actually, when you have that many glucose molecules, in fact, if you wanted to actually physically have a mole of glucose in your hand, how would you do it? Or if I wanted a mole of sodium chloride, mole of table salt in my hand, how would I do it practically?
You have to do the AMU, you have to do the mass. So
Yeah, we go on the periodic table and we would look at the atomic weight for the, for the particular molecule, and that's going to be the grams per mole. So we just lay out that much. If it was salt, it's like 58.5 grams is 1 mole of table salt. So is that someone had their hand up?
Oh, I was gonna say you convert it to grams. You said it.
Say
I
it
was
again?
gonna say you convert moles to grams to figure out how much a mole would be, but you already said it.
Yes, and the way to convert from moles to grams is using that atomic weight from the periodic table there. So you should have basic skill in that. We'll talk a little more about that. So moles of solute per liter of solution is molarity. It's important to remember that it is a concentration, right? It's a ratio of solute per amount of solution, right? Because I could have like the classic thing, like the ocean. The ocean has a particular concentration of salt. And if I take a cup of ocean water, it has the exact same concentration as the entire ocean. But there's way less salt in a cup of ocean water than in the entire ocean, right? So it's not about the sheer amount of solute. It's about what is the ratio of the solute to the volume. So molarity, we'll be using molarity a lot in the class. You want to, and we'll often the molarity is going to be small. So like if I have 0.15 molar NaCl, I might instead say 150 millimolar NaCl. Right? Millimolar is just 10 to the minus third moles. So these are the same thing, right? I'll be using millimolar or molar interchangeably. Just the same way you would say, oh, this thing is 150 millimeters or it's 0.15 meters, right? It's just metrics. All right. Let's talk about osmolarity, which is going to be critical in this class. This is something, so molarity, I am assuming you have seen and worked with quite a bit from chemistry. And if it doesn't seem as, if you're trying to feel more comfortable with it, you should go back and review it. But I'm assuming you all have basic skills understanding molarity. Osmolarity you might not have seen, but it's going to be really important for our class. So osmolarity is similar but fundamentally different from molarity in that instead of looking at the moles of some particular solute, like just looking at glucose or sodium chloride or whatever, it's looking at the sum total of all the solute particles in the solution. And when we're thinking about osmosis, it doesn't matter what the particle is. It doesn't matter if it's a sodium ion or a glucose molecule. All that matters for osmosis and which way water is going to move is how many things are dissolved on one side of the membrane versus the other. So for osmolarity, it's going to be moles of total solute particles per liter of solution. So changing that word solute to like total solute particles actually does make a big difference. For example, when we think about dissolving electrolytes versus carbohydrates in water, say I have I could have 2 molar glucose. This means we have 2 moles of glucose per liter of solution. 2 molar NaCl is 2 moles of NaCl per liter of solution. So both of these are molarity. Both of them are 2 moles of whatever this solute is per liter. If I want to think about the osmolarity, though, it's going to get a little trickier. What is going to be the osmolarity of my glucose solution here?
It'll be particles, so it'd be 12 plus 6 plus 6, 24, 2 make 2 moles of, so 48.
No, so it's important to remember when you dissolve a covalently bonded thing like glucose, it does stay intact. You don't actually, it doesn't dissociate or fall apart. So every individual glucose stays intact as a little intact little glucose. So the way I like to do this kind of thing is called dimensional analysis. We could say, all right, we know we have 2 moles of glucose per liter. What's the conversion? We want to have solute particles, moles of solute particles per liter. So what is going to be the conversion? Every mole of glucose is going to be how many moles of particles?
Well, aren't you going to take carbon on the periodic table?
So you're getting too complicated.
Oh, okay.
Just one, because it doesn't dissociate.
Right, so exactly.
Wow.
So because if you were trying to get like the grams per mole or something, then
yeah,
you would
yeah.
have to do that.
Okay.
But this is because every glucose stays intact as an intact particle, an intact glucose in solution. If I have a mole of glucose, I have a mole of particles in the water. So this is 1 mole of glucose is the same as a mole of particles, so the moles of glucose cancel out, and this is just going to be 2 osmolar, 2 moles of particles per liter. Does that make sense? What about with the sodium chloride, though? If I take sodium chloride, what happens to every mole of NaCl that goes into the water?
So they separate.
It separates into 2 independent particles. We're going to have every NaCl becomes Na on its own and Cl on its own. So how many moles of particles are we going to end up with here?
4.
2.
There.
Oh, so in this one, we're going to have 2 moles of particles. And yeah, when you do the math, this is going to be 4 osmolar. So this is another reason why it's critical to know whether or not something is ionically bonded or covalently bonded. Because the relationship between molarity and osmolarity is very different in these electrolytes. And again, we're going to talk about in a little bit the idea of osmolarity, and for things to be in the body, for a blood cell to be happy in your body, the osmolarity surrounding it has to be 300 milliosmolar, more or less. If you are talking about dissolving glucose or dissolving salt, the amount of glucose or salt you dissolve to create a particular osmolarity really is going to depend on whether or not it is dissociating or not. So you're going to have to pay attention to this in some of the, in fact, quite a few of the calculations we're going to be doing down the road. When you're trying to figure out what is the osmolarity of some particular solution, it's going to depend both on what is the molarity of the original solute, but then it will also depend on whether or not that solute dissociates and either doesn't or does increase the osmolarity. So osmolarity important. And we'll be doing this a bunch. Let's do one last little thing just to complete our discussion of concentration, then we'll take our break. So most of the stuff we're going to do in the class is going to be talking about molarity or osmolarity, in chemistry as well. But obviously, in a practical world, it doesn't make sense. You can't. In fact, you have to convert things to grams in order to weigh them out to make a particular molar solution. So often in the real world, in the practical world, rather than specifying things in molarity, you specify them in what we call percent solution, just like the weight. How many grams of solute per grams of solution? So let me just complete our discussion of concentration by looking at percent solution. Percent solution. It can be by weight or by volume. And it's basically grams solute, grams of solution. So this is much easier. You don't have to convert anything from moles to grams. Grams you just measure out on a scale. And it's actually pretty easy to make. Obviously, it can be also like mils of solute over mils of solution. If it's by liquid or by volume. One of the nice things, if you just want a quick and dirty, let's say I want to make a 4% glucose solution. I should say this is times 100 because it's percent. So grams of solute divided by grams of solution times 100 would give me my percent solution. Water. How much does water weigh? If I have one mil of water, how many grams is that? Anybody? Yeah, 1 gram. Exactly, it's 1 gram. If you want to kind of just basically review, let me just. 1 milliliter, 1,000th of a liter. It's also the same thing as 1 cc, 1 cubic centimeter. Right? If you take a centimeter cube, the volume of that cubic centimeter is a mil. So like if you're measuring out volumes, you could either be cc or mils. It's the same thing. It's just 2 ways to say the same thing. And weighs 1 gram, which is nice. So if I take 100 mils of solution of water here, and I dump in 4 grams of glucose, I'm pretty much done, right? Because 4 divided by 100 is 4%. And because in this 100 mils is 100 grams. Obviously not quite, because adding the 4 to this is going to be, change the weight slightly. But in general, percent solution is used because it's much more practical and easy. And you'll see things that are specified by percent solution all the time. We'll do a little bit of exercises with percent solution just to get you a little more comfortable with it. But I just, you should know that one as well. When you see percent solution, it's just another way of specifying concentration, except instead of going into moles, you stay in the world of grams. All right, any questions about any of this stuff? All right, so what we're gonna do now is take a break. All right, so welcome back. I really, one thing that might be useful to kind of tie things together with all the things we're talking about with osmolarity and kind of make it practical. I like to do this to show like why we care. Why do we care that sodium chloride dissociates into two ions instead of staying as a single entity? And why do we care that molarity and osmolarity are sometimes the same and sometimes not the same? Like these are IV flush syringes. This is like if you have somebody with an IV and you want to like clean out the line, you're going to take this and you'll jack it in. And this is salt water, this is saline. And you want to make sure as this saline goes up into these people's veins that you're not going to destroy their blood cells. And again, blood cells are red blood cells, RBC. Pretty much all of your cells have an osmolarity. The dissolved stuff inside the cell is about somewhere around 0.3 osmolar. You look at all of the sugars and ATPs and proteins and everything, and RBC, any of your cells is about 0.3 osmolar, which means that your plasma is also 0.3 osmolar. So pretty much your most physiological fluids have to be about 0.3 osmolar, or else your cells are either going to, what would happen if the osmolarity is too low outside? If I have like way more stuff in the cell than outside the cell, where does the water go? If I have a lot of dissolved stuff in and not much out, the water will go in. We have like things, we're going to talk about this in way more detail. You have like a hypotonic solution. If it's too dilute out here, the water comes in and the cell inflates or explodes. If it's too much stuff, too salty out here, then the water gets sucked out, kind of like putting salt on a slug. All the water gets sucked out and the thing will shrivel up. So it's important that the fluid outside the cell matches the same osmolarity as inside the cell, so we're in balance. If we're not balanced, the cell is either going to shrivel up or it's going to inflate and explode. So if I am going to inject this stuff up into somebody's vein in order to flush out the IV line, this had better be 0.3 osmolar. Otherwise, my red blood cells are going to get stressed out. So, and this is saline. This is a saline flush syringe. This is salt. So my question would be, what percent saline, NaCl, should I use for IV flush? And I'm trying to put this in a kind of more of a practical context of why this matters. So what's gonna be our starting point? What's the only piece of information I really know that is gonna be a defining quality of the solution that I'm setting in?
0.3 osmosis.
It's gonna be 0.3 osmolar. So that's gonna be my starting point. I'm gonna be, starting. I know it has to be 0.3 osmolar. So, and by definition, 0.3 osmolar means what?
3% or 30%.
Gonna go back to the definition that I was drawing.
Take 3 particles per 100.
So 0.3 moles of particles per liter of solution. So I can start with 0.3 moles of particles per liter of solution. And now I need to somehow make it into percent saline. So the next thing I'm going to do here, I have moles of particles, but I want to have it be sodium chloride. So the first step here, I can use this dimensional analysis. What would be a conversion that's going to start bringing us closer to this? Instead of moles of particles, if I wanted to be moles of sodium chloride, what would be the conversion factor?
Liters over.
Does that matter?
Be liters over something.
Yeah, so we want to eventually have moles of NaCl instead of moles of particles. What's going to be the relationship of moles of particles to moles of NaCl? We talked about this just before the break. It's going to be 2, right? Because every mole of NaCl becomes 2 moles of particles because it dissociates. So that's going to be this next step here. So, and again, in this dimensional analysis, you just come up with a conversion factor and you make sure that the right things are on the top or the bottom so the relevant things are canceling out. So now we have moles of NaCl per liter. We want it to be percent saline though, so that means we have to bring it into the world of grams. So instead of moles of NaCl, I now want to have grams of NaCl versus moles of NaCl. What's the conversion here? How do I figure out that?
Atomic weight.
Yeah, using the atomic weight. And if we go into the periodic table, it's just going to be 58.5 grams of NaCl per mole. And again, if I'm confused about what goes on the top or the bottom, I just make sure that things are canceling properly. So now the moles of NaCl cancel out. So at this point, I have grams of NaCl on the top and liters on the bottom. If I just multiply through here, I don't know if anybody has like their iPhone with a calculator on it right now. Like, what do we end up with?
Eight 0.5 minus 3,
What's that?
17.55, 58.5.
8.775.
Say it, Michael?
8.775.
So basically 8.8 grams NaCl over 1 liter. And then percent saline is going to be the grams of NaCl over the grams of solution. So 1 liter weighs how many grams? 1,000, right? 1 mil is a gram. A liter is 1,000 mils. So this is going to be 8.8 grams over basically 1,000 grams. And then to be percent solution, I have to multiply times 100. And it basically becomes 0.88, which is basically 0.9% NaCl. Does that make sense? How I got there? And just to, so and then if we look I can hand these out. I don't know if you can like. I mean, if you read, what do they say on there? In terms of percent
0.9.
saline,
0.9%.
they say 0.9% saline. Like I'm going through this because I want you to see it's not just random saline in there that they're injecting up your veins. Somebody has spent the time to figure out and doing the math, figuring out what is the percent saline that is gonna be 0.3 osmolar, because that is what is going to keep your blood cells happy and keep them from inflating or shriveling up. All right, so I'm trying to just make it clear that this matters. This idea of osmolarity, this idea of keeping track of this is not just some kind of intellectual exercise we're doing in physiology class. It's something that is very relevant to how do you make sure you take care of somebody and not like explode their blood cells on the, on the floor. So anyway, just kind of putting that out there, trying to give it a little more context. And we'll be spending a lot more time as we go on looking at osmolarity, looking at hypotonic, isotonic solutions and all that.